Alright, hi everybody, this is Jubilee again, and welcome to another TA review. So remember last week we learned about two of the big four tissue types. We learned about epithelium and connective tissue. So this week we are going to be adding on another tissue type, so that's going to be nerve. So the peripheral nervous system is what we're going to be learning about. And then we did cover connective tissue last week, but we did mainly general connective tissue. And remember, when we learned about connective tissue as a tissue type, it does have some specializations within it, right? So some of the specializations of connective tissue are cartilage and bone. Another one is blood, and we'll learn about that another week just because it is kind of a big topic. But this week we are going to be learning about some of those specializations of connective tissue, cartilage and bone, as well as one of the other four main tissue types, nerve. And we are just going to be focusing on the peripheral nervous system, not the central nervous system, so not like your brain or spinal cord, just because it does get kind of complicated. So we're just going to be focusing on the peripheral nervous system. Alright, and by the end of this PowerPoint, you should be able to identify all of these things. You should be able to tell me what these are. But yeah, let's get started. Okay, so we're going to start with cartilage and bone. And again, this is a subtype of connective tissue, one of our big four tissue types. Okay, so cartilage has a lot of functions, but one of the things that it does is it is an embryonic blueprint for bones. Okay, and what that means is whenever you are a fetus and you're still being formed, and your bones haven't formed yet, what happens is the fetus lays down a little blueprint of the bone that is completely made of cartilage. Okay, so if you ever see a fetus or anything like that, they actually are, they're very soft, they're very squishy and flexible. And that is because their bones have not fully formed or ossified yet, so they are mainly cartilage. Okay, so that is going to provide the blueprint for bones, and we're going to learn later on in this PowerPoint how that works, how we transfer this cartilage blueprint to fully formed bones. Okay, and then it will also remain across some of the surfaces of your bones to help form joints. Okay, so there will still be some remaining cartilage at the ends of those bones, and that does help form joints because cartilage creates this very smooth yet tough surface for joints to be able to slide across each other, so for the bones to be able to slide back and forth to create this really nice joint to allow for mobility. Okay, and then it also acts as a cushion, so especially in those joints, right? So we want those joints to be tough, right? We want your bones to be tough and everything, but if you like jump or something, we don't want the bones to just completely shatter because they're so tough. So what we have on the ends of those bones in those joints is cartilage again, right? And it has limited compressibility, and so that does help to cushion some of the blows, so that if you're jumping or up and down or anything like that, there is that limited bit of compressibility in that cartilage to kind of help with shock absorption. Okay, and then we're going to talk about bone. So bone, obviously, it provides skeletal support to land animals, especially, right, because we are bearing a lot of weight. We're not in water or anything, so we're heavy, and so we need a strong, tough substance that can support our weight out of water, right? So bones are really helpful for that. It also encloses and protects our vital organs, right? So not only does it provide support, it helps us stand upright, but it protects our vital organs, so our rib cage and our skull, right? If you think about our vertebrae, so our rib cage is going to protect our heart, lungs, right? Skull is going to protect our brain, obviously. Your vertebrae is going to protect your spinal cord. So it encloses and encapsulates those very sensitive organs, just so that they don't get damaged or anything, right? Because bone is very tough. And then it is also the location of hematopoiesis, okay? And hematopoiesis is just the process of forming white blood cells and red blood cells, right? So in some of our bones, they are designated for hematopoiesis, and so that is a big important site in our bones. Okay, so we're going to go over the types of cartilage. And so there are 3 big types of cartilage that we're going to learn, and these are them. So the first one is going to be elastic cartilage. So elastic cartilage, it's just like what it sounds like. It's meant to withstand stretching, right? It's very elastic in all directions. Okay, so it's kind of like a rubber band. And so the example here that is elastic cartilage is going to be this one to the top left. And it does contain elastic fibers. So you can see in here this kind of darker pink, these kind of darker pink strings that are stretching and connecting all throughout here. Those are those elastic fibers, and that is what allows this cartilage to stretch and flex every which way and be really flexible. Okay, and so when we think of places that's going to be like the pinna of your ears, so the tips of your ears where you would get a piercing. If you feel it, you can tug on it, you can pull it, it'll bounce back. It's very stretchy, it's very elastic, so that is elastic cartilage. The tip of your nose, right, you can squish that around, that would be elastic cartilage. Okay, fibrocartilage is going to be the next one. And so fibrocartilage, it's just like what it sounds. So it's kind of a mix between like what we learned in connective tissue with our normal fibrous tissues and cartilage. So it's kind of an in-betweensy one. And so it is very fibrous, it is very tough, and it is meant to withstand pulling forces in one direction, right? And so that the example of that is going to be tendons, right? Because we said tendons and ligaments are only going to be pulled in one direction, right? Tendons are very tough, and so they are made of fibrocartilage. And a way you can tell it's fibrocartilage is because all of these fibers are running in the same direction, right? Just like they would in a tendon. Okay, and then you can see the nuclei. So these are the nuclei of those chondrocytes in that fibrocartilage, and you can see they're all kind of stacked up on each other, right? They all form a neat little line. Here you can see another one, a neat little line where the nuclei are all stacked up, very organized in the same direction. So that is very characteristic of fibrocartilage. Okay, and then the third one, the one that is kind of the most common and most generic, is going to be hyaline cartilage. Okay, and so hyaline cartilage, it's very characteristically this kind of purpley color, and it's got these chondrocytes in here. So chondrocyte, all of these are going to be chondrocytes, the main cells within cartilage, and that literally means cartilage cell, right? Chondro, cartilage, site, cell. So it's going to have these little chondrocytes in their little lacunae, and then the hyaline matrix around it. It's always kind of be that very purpley color. Okay, and again, that is the one that's going to provide that blueprint for bones. Okay, so we're initially going to have that hyaline cartilage blueprint that's going to be replaced with bone as we develop. It's also going to be the kind of cartilage that stays there and maintains joints. So it's going to stay there on the ends of bones. And then it's also going to make certain other structures like the trachea. Okay, so this is actually a depiction of the trachea, the hyaline cartilage within your trachea. Okay, so now we're going to do some practice. So what type of cartilage is this? And I will give you a hint, tell you these are the same ones. And it is kind of tricky, so hopefully you're guessing this is fibrocartilage. I know that's tricky because this one is very purple, and this is Dr. Johnson's beloved example of fibrocartilage because it's purple and everybody thinks of hyaline cartilage when they see purple, but this is just a special stain, and that's why it looks like that. Okay, but you can see the reason I know it's elastic cartilage is because it's got all of these elastic fibers weaving in throughout here. Okay, all these elastic fibers. This is a special stain that stains for those elastic fibers, so they'll be that very dark black color. And so you can see all those elastic fibers weaving in throughout here. That's what makes this so stretchy, so flexible. And so this is elastic cartilage. Okay, this next one. So hopefully you can tell what this next one is. It is that very characteristic purple color, and this time I'm not trying to trick you. So this one is hyaline cartilage, and hopefully you can tell. So this is the zoomed out picture. So this is actually a fetal elbow joint. Okay, so it's a little humerus and then a little ulna kind of forming here. So they're still forming and everything because this is the fetus, right? And remember we said that hyaline cartilage is the blueprint for bone. So this is going to eventually be a full-blown humerus, a full-blown ulna. It's just not converted into bone yet. So it actually is the majority of it is hyaline cartilage. Okay, and then here you can see there's that hyaline cartilage, and this is actually from the transition site. So this is the hyaline cartilage turning into bone, and we'll go over that in a little more detail in a second. Okay, this next one is going to be that third type of cartilage, the last one we haven't talked about. And so you can see here, right, we've got all these nuclei stacked up in a row, nice and organized. Everything's going in the same direction. Same with this one, right? This is more of a diagram or a depiction of what it should look like. So we've got all these nuclei stacked up all in the same direction. It's very fiber dense, and so hopefully that tells you this is fibrocartilage, right? Fibrocartilage in here. So that's in our tendons. Okay. So now we're going to talk about that process I was talking about, the process of turning hyaline cartilage, the blueprint for bones, into actual ossified hard bone. Okay, and so the process of that is called, sorry, that's my dog. The process of that is perichondrial ossification. Okay, perichondrial ossification. And so what happens is hyaline is that little blueprint, so that's what gets made originally as a fetus, and then it eventually gets laid down as new bone as the fetus begins to develop. And actually, once the fetus is born, right, once it's a neonate, a lot of that cartilage still remains and still needs to be replaced by bone. It's not nearly as much cartilage as this, but a lot of the ends of bones and stuff like that, there's still a lot of cartilage remaining, especially in your joints and stuff like that. And so I do tell people this is why babies are so squishy. And this is why, like, if you're born and your head is a little funny shaped or whatever, those babies can wear those super cute helmets, right? And so it kind of reshapes the shape of their head, and it is because their bone is still being formed and laying down and finalizing that shape. So their heads are really squishy. That's why you have to protect them and everything, right? That's why they're so flexible. All their joints are super mobile, and it's just because they are they've got a lot of cartilage in them. Okay, so it's not quite as tough as bone. And there are many steps to how this is ossified, and so we are going to go over those. So these are the steps of perichondrial ossification. It is a lot of words and stuff, so the way that I remember it, or the way I did at first, was real people have careers. Okay, real people have careers is that kind of acronym for that. And so the way I remember that, so the real, the R in there, is going to be this kind of top one, and so that is going to be the zone of reserve or resting. Okay, the R, real people. So the zone of reserve or resting, and the way I identify it is these cells are resting, right? So they're just chilling out here. So you can see these little chondrocytes. This is just the generic hyaline cartilage. They're not organized, right? They're just kind of spattered throughout here. They're not organized or anything. They're literally just chilling. They're resting. They're doing whatever they want. And so that is the zone of reserve. If you see down here in our next layer, real people, so that is going to be our zone of proliferation, our zone of proliferative chondrocytes. And so this is where they're starting to get serious about things. Okay, they're like, okay, we should probably get up, we should probably get ready. And so what they do is they start dividing and they proliferate, and they'll do that in these nice little neat stacked rows. So you can see they're starting to line up, right, and get organized and proliferate. So that is our zone of proliferation. That's how you know when that starts. Okay, so it's around right here. The next one, real people have. So have is going to be our zone of hypertrophy. So this is where the chondrocytes are starting to hypertrophy. That means they're starting to get bigger. Okay, so this is where they're starting to get turn into big boys and stuff like that and grow up. And so you can see they're taking up more space. They're actually eating away the hyaline matrix around them to make more space for themselves. So you can see they're kind of swelling up. You can see their little nuclei in there, and then the little space around them that they're taking up. Okay, so that's our little zone of hypertrophy. And then after that, once they're all grown up and they're big and ready, they will get turned into bone. And so that is going to be our real people have careers, and that's going to be our zone of calcification. Okay, so calcification. So that is where these chondrocytes, so cells of cartilage, are actually replaced by bone. So they're ossified and calcified and replaced by new bone formation. So that's our zone of calcification. Okay, so now we're going to do some practice in the layers here. So we'll start over here, and you can tell, so these chondrocytes aren't very organized, right? They're just chilling, they're spattered all throughout here, so they're resting. So this would be our zone of reserve right here, our zone of reserve, right? Our next one, real people. So here you can see, right, they're starting to line up, they're starting to get more organized, they're proliferating on top of each other. So this is going to be our zone of proliferation, okay, our zone of proliferation, where they're starting to get organized. Here, they're starting to swell up, right? They're taking up more room, they're getting bigger, they're getting more mature. That would be our zone of hypertrophy, real people have, okay, our zone of hypertrophy. So that's going to be this zone right here. And then now you can see we're starting to eat away at all of that hyaline matrix, and it's being replaced by bone. So this zone right here would be our zone of calcification. Okay, so that's where everything is finally getting ossified into bone, and it's making that hard matrix that we'll cover in a second. Okay, so now we're going to cover bone. So we went over cartilage. So now we're going to cover bone, and in bone you mainly have two different types of bone. Okay, so the first one is going to be spongy bone, and that's mainly in the middle of your bones. Okay, the middle of your bones, and so this is the site of hematopoiesis in long bones. Okay, so this is where you have all of those precursors to all your white blood cells, red blood cells, platelets, everything like that. That's where everything is being made, in the center of bone, okay? And then you have these little like bony spicules and stuff like that that provide structure to the inside, to the spongy bone. And so those little bony spicules in the spongy bone, that's the actual bone content. It's constantly being broken down and built back up to meet the needs of the body, because remember, your bone is a big storage site for calcium. And so you're storing calcium, and so if you are in need of more calcium or anything in your blood, it will actually break down those little parts of the bone. And then if you eat a bunch of calcium and you don't need it, it will build those back up to store them, okay, and keep them in there. So it's constantly being built and broken and built and broken, and that's kind of the cool thing about bone, is it has a high turnover rate, okay? And in the spongy bone, you have these three different types of cells that we're going to learn about. So it contains all three. So osteoclasts, osteoblasts, and osteocytes, right? So the term osteo refers to bone, right? And then the latter term is going to tell you kind of what they do in the bone. We'll cover that in a second. And then the other type of bone tissue that we'll have is going to be compact bone, okay? And so both of these can be in the same bone. They often are in the same bone. They're just different types of bone within there, okay? So compact bone, it's just like what it sounds. So it's very compact, it's very dense, it's very tough, okay? So it is the outer protective shell of bones. It is very structurally sound. It's almost amazing how tough that part of your bone is. And because it is so tough, so structurally sound, blood cannot pass through it, okay? Because it is filled with calcium and osteoid that we'll learn about. So it's very tough. And so in order to get blood to these cells, right, so we still need to get nutrients to these cells because they are still alive. We have this whole special kind of system to get blood to every single cell in here, because blood cannot freely diffuse throughout here like it can in your soft tissues. So we have this special nutrient delivery system called the Haversian system, and we'll learn about that in just a second. And then it does contain osteocytes, okay, so one of those bone cells within it. Alrighty, so first we're going to look at the histologic view of spongy bone. And so I did tell you, it contains these three big different kind of bone cells, right? So osteoclasts, so it means bone, and then clast, I kind of think of catalyze, right? So breaking down bone. So that is what osteoclasts do. They break down bone, right? And remember I said that's constantly happening. If you need more calcium, they're going to break down bone. And so that's not a bad thing that we're breaking down bone. It's just releasing that calcium, okay? So osteoclasts are going to catalyze, they're going to break down bone, and the way they can be identified is they are multinucleate, okay? So if you look at this cell right here, it looks like it's like 5 cells or 4 cells or something, and they're all squished together, right? But really what it is one huge cell that has multiple nuclei, okay? Multinucleate. All right, and so that is very characteristic of an osteoclast. If you see a big multinucleate cell like that, that's going to be an osteoclast, and that is going to be breaking down bone, okay? The next type of cell is going to be an osteoblast, and the way I think of that is osteoblasts build bone, blast build, and so they are going to be laying down new bone, right? So the clasts are going to be breaking it down, the blasts are going to be building it. They're constantly working against each other to remodel this bone, okay? And the way you can identify them is they're constantly going to be laying down new bone on the surface of already existing bone, okay? So this pink stuff right here, this is osteoid. This is already existing bone matrix, okay? And so these cells are lining up on the edge of that because they want to build new bone. They want to lay down new bone. So if you see a cell that is right on the periphery of that osteoid, and it's not a giant cell like this, it's probably an osteoblast, okay? So these are going to be your osteoblasts. They're going to line up on the edges of bone to lay down new bone. Okay, so that's osteoblasts. And they're going to form more osteoid, right? So osteoid is just this kind of pink, tough stuff that comprises the extracellular part of your bone. The next one is going to be osteocytes. And the way I think of that, so it really means bone cell. That's what cytes mean. But the way I think of it is they chill. So osteocytes chill. Okay, and so basically what that means is it's a metabolically inactive osteoblast. So these osteoblasts are constantly building down new bone, laying down new bone, and if they do such a good job at laying down new bone, they actually trap themselves in that osteoid that they're making, right? So if they lay down so much bone around them, they kind of paint themselves into a corner, they're trapped in there. So they can't lay down any more bone, they've already done enough. And so then they kind of become an osteocyte. So they become metabolically inactive and they chill. So they just chill in the osteoid. They maintain the little osteoid around them, but that's basically what they do is they chill. And if an osteoclast comes and breaks down all this bone and releases this osteocyte, it can reconvert itself back to an osteoblast and start laying down new bone. Okay, but once they're trapped in there, they just kind of chill. And then the other thing that is in the spongy bone, right, we said this is the site of hematopoiesis. So we're not going to go into too much detail, but if you're going through all these cells and you're like, well, what's this one? Well, what's this one? Well, what's this one? Honestly, I couldn't tell you. Okay, I could maybe identify some of these, but I'm not going to ask you to identify some of these because most of them are progenitor, white blood cells, red blood cells, everything like that. They're stem cells, okay? So they're precursors to other white blood cells and stuff like that, okay? So you don't need to know them because they're very hard to differentiate anyways. Just know that they're hematopoietic cells. They're kind of doing their thing in there. Okay, so here you can see, so again, right, this is that barrier where we're changing from hyaline cartilage to bone, and so you can see there's a lot of activity going over here. So we're going to do some practice and try to identify those 3 big types of cells. So here you can see this cell, right? So it's not 3 different cells, it's actually one big cell with multiple nuclei within it. So that is going to be an osteoclast, right? Because remember, osteoclasts are multinucleate. So that is going to be a multinucleate osteoclast. And so remember, that's going to be catalyzing bone, breaking down bone. It can also break down cartilage. So that's what it's actually doing here, is it's breaking down the cartilage. But that only really happens, when we're turning this cartilage into bone. So when you're developing as a child or a neonate. Okay. And then here is another example of an osteoclast. So here you can see it's a big cell. It's got multiple nuclei, right, all kind of clumped together. So that's another osteoclast there to break down bone and cartilage. Okay, if we look for an osteoblast, you can see here, right, there's a bunch of cells lining up on the outside. Here's a bunch of them, right, all lining up on the outside of that osteoid, okay, laying down new bone. So those are going to be our osteoblasts. They're building new bone. And then here you can see, right, so this is a cell. It's trapped in the osteoid around it. Here's a cell trapped in the osteoid around it. Here's another one trapped in the osteoid around it. Those are going to be our osteocytes. Okay, here's some more osteocytes up here, but this is actually compact bone. So there's some more osteocytes up there. And then remember, they're just chilling, maintaining the osteoid around them. Okay, so now we're going to look at compact bone. So compact bone is a little bit complicated, and remember that's because we said we've got so much osteoid in here, right? It's very compact. We've got that very hard osteoid that we actually can't get blood to freely diffuse through it like we can for other soft tissues, okay? So we have this special system called the Haversian system. And it's basically just the way that bone organizes itself in order to allow for nutrients to get to every single one of these trapped osteocytes, okay? So some of the components of that are going to be the Haversian canal. The Haversian canal is kind of this main big circle, and blood actually flows through here. So these main big circles in the middle of these little Haversian systems are filled with blood, and it goes in an up and down direction through the bone. So it's actually coming out at you in this view because we cut it at a cross section. So it's actually coming out at you. And so that is going to be kind of the big central supply of blood to each of these little Haversian systems, right? So here's one, here's one, here's one. So that's going to be the main supply of blood in the center. Another thing we have is a Volkmann's canal. So the Haversian canal are the main ones in the center, but in order to connect one Haversian canal to another, we have these things called Volkmann's canals, and they go transversely. Okay, so they connect each other transversely. You just can't see one every time you cut a cross section because, again, it's just the way that you cut it. You have to cut it very specifically and very perfectly in order to be able to see where it actually connects them. Okay. Another thing you have is lacunae. The way I think of lacunae is it's like a little cocoon, so lacunae, cocoon, and so it's basically just the space in the osteoid, right? So it's the space in that dense, hard osteoid that the osteocyte is sitting in, okay? So the osteocyte is chilling in its little lacunae. And so here you can see, so this is a special stain for bone, and so you can see each of these dark spots here are osteocytes, okay? And you can't really tell or really see, but I know if that's an osteocyte, it is sitting in a lacunae. It is sitting in a space in that osteoid, okay? So people kind of get confused between the difference between those just because it is very hard to visualize, but it's basically just the space, the very small space that the osteocyte is sitting in. The next thing is going to be the concentric lamellae. So that basically means like within a circle, right, around the center, and lamellae is layer. And so you can see these are kind of like tree trunks, right? So they've got little layers around the center. So concentric layers around the center, concentric lamellae. So that's basically what it means. It just means the way that this is set up like a tree trunk. There's multiple layers in it, okay? Another one is going to be canaliculi. And so remember, we've got blood in the center through here, and this blood in the center needs to get all the way to the outside of this Haversian system, to this little osteocyte. And so the way that works is we've got these little tiny strings, so these little black strainy things that you see radiating out of the osteocytes, those are canaliculi. So they're just connecting little microscopic tunnels that connect this Haversian canal to this lacunae with the osteocyte in it, to the next lacunae with the osteocyte in it, to the next one, to the next one. And so that allows for blood from the central Haversian canal to get all the way out to the outermost osteocyte in that system. Okay. So here you can see it a little better with a little more detail, and there's a little diagram. So we've got this central Haversian canal, right? So there's going to be blood in there, and it's going to radiate out through these layers, through these lamellae, and it's going to do that through the canaliculi. So here you can really see these black little stringy lines, right? These black little lines. Okay, those are those canaliculi, and they're connecting the central Haversian canal to this lacunae that contains an osteocyte, to this lacunae that contains an osteocyte, and so forth. Okay, so that's just kind of what's happening, and you can see it a little better in the diagram. Okay, and so here is kind of a better way to see large scale how everything's set up. So this is going to be the compact bone on the outside layer of that bone, right? And inside we've got spongy bone and whatever. But here you can see, right, we've got all of these Haversian systems, right? And they're all lined up. And in the center are those Haversian canals, right? Haversian canals. And so remember we said to connect one Haversian canal to another, the ones that go transversely and connect them are going to be Volkmann's canals. Volkmann's canals. And so that's why it's really hard to see them, right? Because if I do a cross section here, I'm not going to see any, right? If I do one here, I might see some, but I'm not going to see the whole length of it, right? If I want to see the whole length of it, I have to cut it this way, and that's a very specific and hard way to cut it. Okay, so it's very hard to see Volkmann's canals, but they do exist, I promise, and this kind of helps you understand that. And then here is kind of a real picture of bone. It doesn't have that special stain, so it makes it harder to understand things, but here you can see, right? So this is kind of that Volkmann system with those concentric lamellae, right? They're all radiating out around the center, so that's going to be a Volkmann's canal. And here you can see those little lacunae, so those spaces, and within those spaces are going to be those osteocytes. Okay, so now we're going to move on to nerve, and remember we're doing peripheral nerve. And so nerve is specialized for the transmission, reception, and integration of electrical impulses, right? So it is basically a messenger, but a very fast messenger and a direct messenger, okay? So it does electrical impulses. And so we're going to learn about neurons, right? So that's the main cell of nerve. And so what happens is they're very excitable cells, okay? So what happens is they pick up on a little electrical impulse or something from their dendrites. They have multiple dendrites. That goes to the cell body. The cell body decides what to do with them. So if they're like, okay, we're getting a lot of the same message, this is probably really important, I should probably pass it on, okay? Then they will. If they get like one tiny little message over here and pretty much nothing else, they're like, this probably isn't that important, I won't pass it on. So that's kind of what the cell body does in a very generalized way. And then if they do decide to pass it on, they will send it really quickly, instantly through this thing called an axon. And that's going to go either to the next neuron cell or to the organ that it's going to. Okay, so smooth muscle, an organ like the liver, anything like that. Okay. So that's kind of how that works. Okay, so in peripheral nerve, we have these kind of two terms used, and so the first one is going to be ganglion, and a ganglion basically means a cluster of neuronal cell bodies, right? So it's where you've got a bunch of those cell bodies of neurons all together in a big cluster. So here is a cell body here. You can kind of see parts of others. It's just not a very good cross section, but there's a big cell body there. They do kind of all cluster together, and they talk to each other and stuff like that, and it does kind of form a mini brain, because right, remember I said they're kind of deciding whether or not they should send an impulse through. Not everything gets sent through, because then your brain would be overloaded with stimuli. So they do kind of decide what's important and what's not on what to send through. And then the next term we're going to do is going to be nerve. So nerve is kind of the general term for a bunch of axons. Okay, so ganglion is a bunch of cell bodies clustered together, A nerve is a bunch of axons clustered together, and they all kind of travel in the same direction. So you can see they're all kind of going up and down in the same direction, and they can branch off at similar or different points going to their target tissue. Okay, so we're going to talk a little bit more of ganglion. So here's a better picture of that, right? So we've got a bunch of neuronal cell bodies, and they're very big. Okay, they're very big. So this whole thing is a neuronal cell body. Same with this one. Okay, and you've got a little nucleus and a nucleolus within those. Okay. And so when you look at these neuronal cell bodies, you'll notice they look kind of grainy, right? There's a lot of little spots and dots in here compared to the tissue around them. And so what that is called is Nissl substance. And so when we refer to Nissl substance, it basically indicates a very metabolically active cell. Okay? And so as you can imagine, neuronal cell bodies are very metabolically active because they're constantly sending things, receiving things, transmitting signals. So they are very metabolically active. And so that is why the cell body contains Nissl substance. And basically what that is it's composed of ribosomes, polyribosomes, and rough endoplasmic reticulum. And so that's what makes this kind of graininess. Okay. And remember, polyribosomes, ribosomes, and rough endoplasmic reticulum are very important for like protein synthesis. All right, so it just means we're making a lot of things in this cell. It's a very metabolically active cell. And so we kind of characterize that on the light microscopic level as Nissl substance, that kind of grainy stuff. And then around the cell bodies, we have these things called satellite cells. So you can see here, so this is that neuronal cell body. And you see these cells, they're kind of wrapping around it, right? The cell's wrapping around the neuronal cell body. So that would be a satellite cell. And basically what it is it is a cell that supports the neuronal cell body. It kind of provides structure to it, around it. It protects it and stuff like that. If it gets damaged, it'll kind of help out. So it's kind of like a bodyguard around these neuronal cell bodies because they are very important. So now we're going to talk about axons. So axons are the ends of the neuron that actually transmit the message to the next cell. And so they are kind of like little wires, right? Sending electrical signals. They're sending electrical impulses. So here is an axon, and they can be really long, right? They can go all the way from your brain to the tips of your toes. So they are very long. And so what happens to them a lot of the time is they get covered in this thing called myelin. So it's this thing called a myelin sheath. And basically what that is it's like the rubber that's around a wire, right? And that's there to protect the wire. It's there to enhance the signal so that the signal can travel faster, right? So that we don't have any interference between the electrical impulses and whatever's happening around it. Okay? So it protects them, and then that myelin is made by these cells called Schwann cells. So these Schwann cells will wrap around an axon, and they'll create this thing called myelin to protect them and wrap around the axon, okay? And in between each little Schwann cell, there's a break in the myelin, and that is called a node of Ranvier, nodes of Ranvier, okay? And so what that does is, and we won't get into too much detail, but as this signal is traveling down this axon, it allows for the signal to skip from right here to here. And so it kind of does this skipping thing in between each of the nodes of Ranvier. It makes the signal much faster. And I won't get into all the detail because there's a lot of physiology behind it, but just know that myelin makes the signal faster, okay? And we need those nodes of Ranvier, those little breaks in between them. Okay, and then now we've got, little zoomed in pictures of the axons and their myelin. So here you can see the kind of innermost circle of this is the axon, and then you can see this kind of little coat around it. That's going to be the myelin, right? So this is the axon in the center, the little coat around it, that's the myelin, right? So that's surrounding all of these axons to protect their signal and make things go faster. Here is a transverse section of it, and so you can see in the center, this is a really good example. There's that axon, and then this kind of fuzzy coat going around it on both sides. That's the myelin. And then here you can see there's a little divot in the myelin, and then it continues being myelin. This is a node of Ranvier. Okay, that's that little break in the myelin, right? Then the myelin continues. There's another little divot. That's another node of Ranvier. So you can see the little nodes of Ranvier kind of spaced regularly all throughout here. Okay, and then you can also see some Schwann cells, right? Those are the cells making the myelin all throughout here. Okay, so now we're going to talk about the connective tissue around a nerve, because remember we said connective tissue is everywhere. It glues things together. So it does glue your nerves together, right? To make them all stick together so they can travel in the same direction. So we have different terms for the different types and levels of connective tissue around a nerve. And so we do always kind of use this binary naming system a lot when we're naming things. So epineurium, sorry, that's my dog. Neurium, meaning nerve, right? Referring to nerve. And epi is going to mean like outermost. Okay, so it's the outermost connective tissue around a nerve. Okay, so it surrounds the entire nerve. Okay, so epineurium is going to be this connective tissue that surrounds the entire nerve. Okay, the next one is going to be perineurium. So that's kind of in between these, and it's going to surround the nerve bundles. Okay, so you can see we've got little bundles within here, and so the perineurium is going to surround those. Okay, so it's going to be lining these nerve bundles. And then we've got endoneurium, meaning like innermost, referring to the nerve. So that's going to surround each individual axon. So we have them around each individual axon with its myelin sheath. Some connective tissue. Okay, so here is a real histology example of that. And so you can see this is one really big nerve, right? One really big nerve. And so it has a bunch of connective tissue around it, right? So if you zoomed in here, you could see a bunch of fibroblasts, a bunch of collagen bundles, everything like that. You would see some blood vessels, right? Because blood vessels are in connective tissue. And you can see here, so all this connective tissue surrounding the entire nerve would be our epineurium. Epineurium meaning outermost, right? And then within here, we've got bundles, right? So here's one big bundle, one big bundle, one big bundle, and we've got some other smaller bundles within that, right? So surrounding any of these bundles, this would all be perineurium within here, okay? And then if we zoom in a bunch, right? So in here, in between each one of these, there's very microscopic amounts of connective tissue, okay? You can't really see it, but just imagine and trust that it's there. Microscopic amounts of connective tissue. That's going to be our endoneurium. Endoneurium. So epineurium, perineurium, endoneurium. Okay? And here you can see some Schwann cells. They're like little hats. So some of these are fibroblasts within here. Like this one's probably a fibroblast with the really flat nuclei. But when they look like they're little hats on the myelin sheath, they're probably Schwann cells. And it is hard to tell the difference. Okay, so now we're going to go through some other examples of nerve. We'll cover this more when we cover the skin, but I do just want to show you some examples. So hopefully this is familiar, right? Remember we learned this is all dense, irregular connective tissue. This is that stratified squamous epithelium. And you can see here in this junction between the two, there are these kind of bundles happening, these weird looking things. And so these are actually the nerve endings, the sensors of nerve. And so what they're given a special name, and it's called a Meissner's corpuscle. A Meissner's corpuscle, and they are responsible for light touch. So if somebody just grazes your arm, your Meissner's corpuscles will pick up on that and transmit that to you. Okay, this one is an example of a Pacinian corpuscle. So this is deeper within the dermis of your skin. So this is deeper down. And you can see they look like onion rings to me, right? They've got a bunch of little layers in here, right? So they look like little onion rings, a bunch of layers. And those are going to be your Pacinian corpuscles. Pacinian corpuscles, those are responsible for deep touch, so pressure. So if somebody squeezes down on your arm, your Pacinian corpuscles are going to pick that up and transmit that. So here are some more examples of nerve in your body. So this is in the small intestine. And so you can see here down at the base of the small intestine, we've actually got some little nervous tissue. And so the one that's going to be kind of on the innermost portion is going to be the Meissner's plexus. And don't get that confused with the Meissner's corpuscle, right? We just went over that. The Meissner's corpuscle is in the skin, Meissner's plexus in the small intestine. Meissner is just a scientist that discovered a lot of different things having to do with nerves, so he has a lot of things named after him. But this is the Meissner's plexus, and then this is going to be the Arbox plexus down here. Okay, Arbox plexus down here. And we'll learn more about these whenever we go over the digestive system. Okay, so here are more examples of nerve, because a lot of the times you guys have trouble identifying nervous tissue. But basically the way I identify it is nerve tends to separate itself off and bundle itself off from the tissue around it. So you can see here, right, in this example, this nervous tissue is kind of bundling itself off, right? It's its own little circle. Here's its own little circle, right? So that is nervous tissue. That's a bunch of axons actually right there, okay? And that's different from a blood vessel or a lymph vessel, right? Because yeah, they are circular, but it's not like filled with actual cells. It's filled with blood or lymph. So that's different than actual nervous tissue, right? Here, we've got a Meissner's corpuscle, and even down here, we've got a tiny little nerve bundle. You can see it kind of separating itself off, right? And then here, this one's kind of obvious. So this is a big old nerve bundle, okay? And it's even got some blood vessels in it. But yeah, there's a big old nerve bundle. Here's some more examples. So you can see they're kind of separating themselves off into little bundles. That is going to be nervous tissue. Here's another little bundle separating itself off. And again, that's different than a blood vessel, right? So you see how this one actually has a lumen, meaning an opening in the middle. This one has a lumen, an opening in the middle. This one doesn't. It's filled with cells, right? But it's still bundling itself off. That's a nerve bundle. Okay, so now we're going to do some practice. So what is/are the functions of cartilage? So A, yes, it is an embryonic model for bones. Remember we said that we lay down cartilage first and then it gets replaced by bone. B, slides across each other easily while bearing weight at articular surfaces of bones. Yes, that is correct, because remember we said cartilage helps form those joints. It's very slippery, it's very smooth, so it allows bones to slide across each other easily in joints. And then cushion, as cartilage has limited compressibility. Yes, remember we said that cartilage is important at the ends of bones in those joints for shock absorption, because it does have that limited compressibility. So it would be all of the above. This one, so we're doing some matching with cartilage types. So A, remember we said for A that those kind of darker black fibers throughout here are elastic fibers. And so this is elastic cartilage, so that does not match. It is not hyaline cartilage that's pictured here, so that one is incorrect. B, so this is in the trachea, and remember we said this one was hyaline cartilage, right, with all that hyaline matrix out here. So that one does not match. These are actually switched. And so then C, we can see here all the fibers are running in the same direction. The nuclei are kind of all going in a little neat line. So that would be fibrocartilage, right? And so that one does match. That one is correct. So the answer would be C. Okay, next. So which structures best match the image? And so A, you can see here, it's pointing to a bunch of axons that are myelinated. And so here it says group of myelinated cell bodies. Remember, we never talked about cell bodies being myelinated. That's the axons. So A would be incorrect. Those are not the cell bodies that are myelinated. B, it's saying cell bodies of ganglion neurons. And so here you can see there's even a little zoom in, and you can see these are very big cells, right? With that grainy kind of nissl substance. So those are the cell bodies. Those are ganglion neurons. So B would be correct. And then C, it's saying satellite cells. So if we look here at the zoom in, so this is a bunch of axons, right? Axons. And so remember we said the supportive cells of axons are going to be the Schwann cells, right? And remember we said the supportive cells of the cell bodies would be the satellite cells. So there are not going to be any satellite cells here where there are a bunch of axons. They would be in here. So that one does not match. So it would only be B. Okay, this one. Which structure does not match both of the images, and it has both underlined and not underlined. So A, both of these should be nerve. So if we look at this one, yes, it is a little bundle separating itself off. Yes, that is nerve. If we look at this one is not nerve, right? So it's circular, but we can see this is actually a little lymph vessel or a venule. And you can tell because inside we've got, lymph or something like that, and then we've got endothelium, that one cell thick layer lining it. So this is actually a vessel. Okay, so that is not nerve. So A, it does not match both images. B, it's saying these should both be fibroblasts, and yes, these are both fibroblasts, because this is connective tissue out here, and so these flat cells out here are probably fibroblasts. C is saying perineurium, and so remember we said perineurium is going to be that connective tissue that surrounds nerve bundles, right? Nerve bundles. And so here you can see, yes, this is the connective tissue, the fibroblasts that are surrounding the bundles. Remember, epineurium would be around the whole thing. So that does match both of them. So the only one that didn't was A. Okay. As always, this is simply a review. There will be more on your quiz, so feel free to use your resources. If you do have any questions, feel free to go to help desk, ask me, ask your other TAs. Hopefully this review was helpful, and good luck on your quiz.